everyone. For learning outcome number 2 of Module 12, we're continuing with the nephron, and now we're moving to the nephron loop as we have already talked about the proximal convoluted tubule. So the next structure would be the nephron loop, and then we will go all the way to the collecting system. So here is your proximal convoluted tubule, and it will end as the renal tubule enters the medulla. And this bend marks the start of the nephron loop. Now the nephron loop can also be called the loop of Henle, and it's going to be divided into the descending limb, which is the one that's going down, and the ascending limb, which is the one that's coming back up. So this means that the fluid in the descending limb flows towards the deeper part of the medulla, and that fluid from the ascending limb flows back towards the renal cortex. Notice how the descending and ascending limbs have a thick and a thin portion. The thicker portion is proximal to the cortex, whereas the thinner portion runs continuous in the medulla. This means that the thick loop is made of simple cuboidal epithelium, and that the thin portion is made up of simple squamous epithelium. So when you hear this, what is this telling you? Well, this is telling you that when you have simple squamous epithelium, that the cells are flat, therefore the water can run freely in this type of epithelium. So there's nothing that holds the water from running from inside the tubule to the interstitial space, or from the interstitial space back inside of the tubule. However, the thin limbs, they are actually impermeable to ions and other solutes. So really just water is what's going through them. Now in the thick descending limb, nothing much happens because it's a simple cuboidal epithelium and there's no microvilli like you have in the proximal convoluted tubule, so nothing much really happens. However, in the thick ascending limb, it will contain an active transport mechanism that will pump sodium and chloride ions out of the tubular fluid. And as a result, the medullary interstitial fluid will contain an unusually high concentration of solutes. This solute concentration is usually expressed in terms of millimoles, and near the base of the loop, in the deepest part of the medulla, the solute concentration of the interstitial fluid is 4 times that of plasma. So 1200 milliosmoles for the interstitial fluid versus 300 milliosmoles of plasma. Therefore, this high osmotic concentration surrounding the nephron loop results in this osmotic flow of water out of the nephron. And where is this water coming from? It's coming from the thin, ascending and descending limbs. We mainly have 2 different types of nephrons. We have what we call the cortical nephron and the juxtamedullary nephron. Like the name says, the cortical nephron will mainly be situated in the cortex of the kidney. Notice how the renal corpuscle is located right at the middle of the cortex. In addition, notice how the nephron loop is shorter when compared to the other type, which is called the juxtamedullary nephron. Notice how the juxtamedullary nephron has the renal corpuscle located a little closer to the medulla. And notice also how the nephron loop is way longer than that of the cortical nephron. In addition, we have way more cortical nephron than we do juxtamedullary nephron. In fact, 85% of the nephrons are made up of cortical nephrons, and just 15% are the juxtamedullary nephrons. Now, because the juxtamedullary nephron has a longer loop, it gives more time for the water to exit through the thin limbs. Therefore, the juxtamedullary nephron is the one that's responsible for forming a concentrated urine because it gives more time for the water to leave the nephron loop. And as such, the cortical nephron cannot produce a concentrated urine because its loop of Henle does not reach the inner part of the medulla. In addition, we have these structures that are called the vasa recta, which are a group of slender capillaries that will absorb the water that's present in the interstitial space and return it into the general circulation. So the net effect is that the nephron loop will be able to reabsorb an additional 25% of water from the tubular fluid and an even higher percentage of sodium and chloride ions. Reabsorption in the proximal convoluted tubule and the nephron loop reclaims all of the organic nutrients, 85% of water, and more than 90% of the sodium and the chloride ions because of that active transport mechanism that's present in the thick ascending limb of the nephron loop. And the remaining water, ions, and all the organic waste filtered at the glomerulus remain in the nephron loop and enter the distal convoluted tubule, which is the next compartment. Next, we have the distal convoluted tubule, which will ascend out of the medulla of the kidney and enters the cortex. The distal convoluted tubule, notice how it passes between the afferent and efferent arterioles. The distal convoluted tubule will differ from the proximal convoluted tubule in three ways. The distal convoluted tubule has a smaller diameter. I know it's a little bit hard for us to notice from these models, but it is a fact. Its epithelial cells lack microvilli, which we can see from the model, and the boundaries between the epithelial cells in the distal convoluted tubule are easier to see than those of the proximal convoluted tubule. And these anatomical characteristics, they will reflect the major functional differences, where the proximal convoluted tubule is involved in reabsorption, while the distal convoluted tubule is involved in secretion, even though it does some reabsorption as well. Now, in order to avoid cell damage, the amount of water and salts in the blood, they need to be kept regulated, and the nephron will be the one that will play a vital role in this. The distal convoluted tubule, which is made up mostly of simple cuboidal cells, is going to be the place where the initial adjustments are going to be made to the filtrate. Remember, the filtrate is the fluid that was initially filtered from the glomerulus, or actually from the blood that's entering the glomerulus. And under the influence of the hormone aldosterone, the sodium is going to be reabsorbed, followed closely by the chloride ion, and then potassium and hydrogen ions are going to be secreted into the tubule, and almost all remaining bicarbonate is going to be reabsorbed. And although the distal convoluted tubule is normally impermeable to water, there are going to be small amounts that may be reabsorbed here as well. Now remember that aldosterone is going to be a corticosteroid hormone, which will stimulate the absorption of sodium by the kidneys, and this will regulate the water and the salt balance. Now let's see how this occurs exactly, because there are specific structures in the distal convoluted tubule and close to the distal convoluted tubule that will function to control the aldosterone that's being released by the adrenal cortex, which are right on top of the kidney, right? They sit right on top of the kidney. Now, the juxtaglomerular complex is this structure that's made up of three components. the macula densa, the juxtaglomerular cells, and the extraglomerular mesangial cells. The macula densa, as we can see over here, they're made up of these 6 or 7 columnar cells that are present right over here in the distal convoluted tubule. Now remember how I said that the distal convoluted tubule will swing around and come between the afferent and efferent arterioles. And the epithelial cells of the distal convoluted tubule are made up of simple cuboidal epithelium. So these columnar cells are specialized cells that are forming the macula densa. And this macula densa will regulate the release of a hormone that's called renin. So renin is going to be secreted and stored in the kidneys, and it will promote the production of angiotensin. Now, angiotensin is a protein whose presence in the blood will promote the secretion of another hormone that's called aldosterone, which is the hormone that we were talking about in the previous slide, that is synthesized in the adrenal gland, and it will have a function in regulation of the sodium ion concentration to control and regulate the blood pressure. Therefore, if you have low blood pressure, aldosterone will promote the absorption of sodium, and this will raise the blood pressure, because where sodium goes, water follows, and this increases blood pressure. The next component of this complex is the juxtaglomerular cells, which will release erythropoietin and renin. We already know what renin does. Erythropoietin is going to be important because it's going to be a hormone that's going to be secreted by the kidneys, and this increases the rate of production of red blood cells in response to falling levels of oxygen in tissues. So if you have low oxygen levels, then you have the secretion of erythropoietin that will increase the rate of production of red blood cells. And these juxtaglomerular cells are going to be located right over here on the afferent arterioles. Now, the last component of the juxtaglomerular complex are the extraglomerular mesangial cells. And they were not represented on the model, so I did draw them here. And they're going to be located in the triangular space between the afferent and efferent arterioles. And these cells will provide feedback control between the macula densa and the juxtaglomerular cells. And why is all this important right at this location? Well, remember that from the distal convoluted tubule, it will go into the next compartment, which is called the collecting system. And from the collecting system, it will go into the minor calyx, major calyx, and then it goes out of the body. So this is why it's important to regulate what's staying in the body and what's going out of the body before it reaches the collecting system. So this is the function of the juxtaglomerular complex. So like I said, the distal convoluted tubule is going to be the last segment of the nephron. It's going to open into the collecting system, which will consist of the connecting tubules that will connect to the collecting ducts that will end in the papillary ducts within the renal papilla. Every nephron is connected to a connecting tubule, and several connecting tubules are connected to a collecting duct. Several collecting ducts will converge and empty into the larger papillary duct that empties into a minor calyx in the renal pelvis. The epithelium lining the collecting system begins as a simple cuboidal cells in the connecting tubules, and it changes to a columnar epithelium in the collecting and papillary ducts. So what does this tell us? It tells us that there is more exchange occurring at the connecting tubules than at the collecting and the papillary ducts. The collecting duct is where the final adjustment takes place before the filtrate leaves as urine. Here, the sodium chloride is reabsorbed in addition to the urea. But what about water? Well, levels of water in the collecting duct are going to be regulated by the hormone antidiuretic hormone, which will act to retain water in the body by enabling it to be reabsorbed back into the blood. So, for example, if you're going to be dehydrated, then antidiuretic hormone will allow these structures that are called aquaporins, which basically mean water channels, to be inserted into the epithelium, allowing water to be reabsorbed back into the blood. Remember, the medulla is usually salty, meaning that there are high levels of sodium and chloride. So if water is given the opportunity to leave, it will. On the other hand, if you're overhydrated, a lack of antidiuretic hormone will mean that the collecting duct is more or less impermeable to water, so excess water can pass out into the urine. In general, urine contains water, urea, sodium, chloride, potassium, creatinine, as well as other inorganic substances, but no protein, no red blood cells, and no platelets.